I'm Chi-Min Ho. I'm one of the moderator, and MG is the second moderator, and we have Kevin Xumin. Who is Xumin? I haven't seen him. Ammon and the sheriff. Okay, so I'm going to stand to talk about because the laser pointer doesn't work. So what I'm going to talk about the nanomedicine. Actually, I'm going to talk about even one step further. What is a personalized nanomedicine? So, oh great, no, but maybe it doesn't work on this screen. Give it a try. Doesn't work. No. And so we talk about the personalized nanomedicine. And this technology, what we can do is we can apply to demonstrating one disease, and we can apply to any other disease. So it's a disease model-free and the mechanism-free platform. So we really don't have to know the details about the mechanism to cause the disease and also how the drug work with that protein. And the first one we talk about when we treat the patient, the effects of dose. Dose is quite important. If you don't do the dose right, and then the drug will be either toxic or will be not very effective. And the most time we're doing now is using the maximum tolerated dose. So in order to get high efficacy, and then we tolerate the dose to treat the patient. And then drug, if we use a drug conjugated nanodiamond, we can significantly reduce the toxicity. Let's take one example. That's a murine experiment for breast cancer. And so this is a control case. And then if these two is doxorubicin at 100 microgram and without the nanodiamond. So it's really not very good compared with the control case. But with nanodiamond, and the efficacy become better. But it's still not very good. So we double the dose. We double the dose if without the nanodiamond, one of the mouse or two of the mouse or all the testing group mouse all died. Okay? And but with nanodiamond, and it survived, not only survived, and it's much better than the control case. So that shows, and we can use high dose and with nanodiamond, and we can get high efficacy and much more lower toxicity. And that's not only the case for this experiment, and it's pretty much true for many of the cases being tested. Okay, and most of the time we will treat the disease not only with one dose, and with one drug or two or three drugs or four drugs together. And that's, we can take advantage of the drug-drug interaction, especially take advantage of synergism. And then how do we do the optimized drug dose combination with high efficacy and low toxicity? Then it become a very tough task. Because if we have 6 drugs with 10 dosage, it's 1 million possibilities. How can we find the best one out from 1 million with a simple way to do that? And let me show you one case. And in this case, we did also in the murine case, and we used 3 drugs try to treat the renal cancer. And use 3 different drugs, single drugs, and the single drug really doesn't work. Okay, the tumor size grows with time, and it's almost the same exactly as the control case, no matter it's a high dosage or low dosage. But if we combine 3 together, and with the proper dosage, the 4-2-2 means the low dosage and high dosage. With the 3 drugs, and we combine together, and we search out from millions of possibilities, it works much better, just compared with control. So first, optimize the drug dose combination, and we can get much better result. And another thing is very important. If you go to all high dose, and actually it doesn't work as good as the right dose and the drug combination. So we do have to face the problem, optimize from the best one out of millions or 10 million possibilities. So that's how, if we know how to optimize that. Let's go to personal case. And every patient is different. So we have to provide the right drug dose to that specific patient. that's one case for the renal transplant. For renal transplant, and the day after transplant, 3 months after that, we needed to keep these 2 markers within the shaded area. Otherwise, the patient might have to go through another transplant. And the doctors use 3 drugs and try to adjust the dosage and adjust the dosage such that we'll be able to do that. And that usually is by titration. So if too high, we go to low. If too low, we go high. So usually you get oscillatory case. And the technology we developed is we basically use a small number of testing points, try to calibrate the patient. So first few data points, we follow the input as the doctor prescribed, and we measure the output. So after few test calibration point, and then we can tell we need to use this green dose instead of the red dose. And if we do that, that's what we get. Okay? So we can really just use a few data points, we calibrate that patient. After we calibration, then we know what is the right dose and the right drug for that patient. And then we can guide the patient into that very, very precise and also very thin region of what we designed. And this technology is very simple and also can be applied to almost any disease. That's what the case we have tested. We have tested infectious disease, cancer, organ transplant, stem cell, and all the other indications. And all of that, we have the cell line test. Red ones have animal test, and these ones we have clinical test. And I was an aerospace engineer. Now I'm working on these problems. That's exactly one of the panelists talked about in the previous session. We really have to work in the interdisciplinary area. And I think we have to be able to give up. Okay? I have to give up what I learned, what I followed the engineering training. I have to give that part which really doesn't fit for the medicine. And I have to learn from the medicine and how do we put the 2 sides of the together. And we can use a very simple technique. We can push to the personalized nanomedicine. Thank you.
So I'll pick up from there. I'm M.G. Finn. I chair the Department of Chemistry and Biochemistry at Georgia Tech. I co-lead the Center for Pediatric Nanomedicine, which is a Georgia Tech Children's Healthcare of Atlanta initiative. And I'm also on the NIH Nano Study section, so I see a lot of science in this area. And Chi gave some wonderful examples of the advantages of nanoparticles and nanomedicine approaches, and let me try to generalize it a little bit for those of you who may be new to the field, and I can take you through this conceptually from a chemical point of view. So nanostructures are essentially, from a chemist's point of view, impossibly large. From a biological point of view, they're small, sort of around the size of the building blocks that we're familiar with of biological function. And so the nanoscience has developed to a point where it's become very exciting for medicine because nanostructures are complex enough to have potentially multiple functions. The really advanced things we think about are functions that are triggered off of other functions. So smart materials, or materials that sense their environment and can respond, in essence, replicating some of the procedures that living systems go through, but in a way that we can engineer. So the many revolutions in chemistry, in structural determination, in synthesis of these kinds of structures, in analysis of what they are, and in how to produce them in quantity, and in their evolution. And I mean that both in a realistic and an intellectual sense. Those are the revolutions that have occurred to bring us to the point where, as a field, we can begin to dream realistically about designing a particle or a rod-shaped thing and think of it as a device that one would think of any medical device at the bedside, something to do sensing, something to deliver a therapeutic, something to report on the success or failure of that therapeutic, and to tell you what to do next, right? So the kinds of problems that become accessible, or at least in principle accessible, in nanomedicine are of supreme importance to pediatrics. And this is why I got excited about coming to this from a completely basic science background. I got excited about the opportunity to contribute to pediatric nanomedicine because devices that can do what I've described have dose capabilities that make safety very easy to engineer, have profiles of diagnostic possibility that are critical for treating children. So nanomedicine is, I think, most excitingly applied to pediatric medicine more than any other field. But of course, the tools that are developed in this field go for adult medicine as well. And the one other thing that I'll emphasize is something that she emphasized as well, is that nano approaches are really platform approaches that are in principle applicable to many different diseases and problems. And so we in the field are in the business really of being, very often have an engineering mindset. We need to know basic science and develop basic insights, but we're in the business of engineering functional things, functional little machines that go and do what they should do. So that's the overall excitement level and level of intersection of many fields that make nanomedicine exciting and really at its infancy. I mean, really at its infancy. So this slide here is what I put together to summarize the kinds of things that we're taking a run at Georgia Tech and Emory and Children's in our tripartite approach. The Center for Pediatric Nanomedicine focuses, you can just read this, but the platform nature of the targets, of the way we approach things, I think is evident here. And different nanoparticles can be very different things, actually. And so I'll talk a little bit about our work in immunology, but there's one in the middle, smart nanosystems for drug delivery, happen to be organic polymer systems that have no biological counterpart in terms of their material that they're made of, but they mimic proteins and lipid systems very nicely. One can steal from biology, and we do that a lot to make functional nanostructures. Nanostructures go from metals to ceramics to proteins and lipids and nucleic acids and all sorts of things. So again, we take from every field we can find to make these functional nanodevices. One more slide is all I have. If somebody, oh, do I have it here? I have no idea how to do that. There we are. So this is just a very quick, obviously very quick thing to what we do. We work with the virus-based nanoparticle area end of the field. So viruses are, in fact, nature's nanoparticles, and they have a wide variety of functions that we can hijack and certainly learn from. And many of the therapeutics and diagnostic agents, and indeed all cell-penetrating peptides, come from viruses, for example. And so we work with viruses, we engineer them, we evolve them. That's another thing we love about nanoparticles, is they are evolvable in the biological sense as well as in the engineering sense. And we do all sorts of things to them, and we send them at all sorts of targets, but our primary emphasis is on immunology. So that's where I'll stop in just a personal introduction and a little bit of an introduction to the field. So now we're going to go to our three distinguished panelists. They're going to give you a little bit of their take on the field, and then we'll get some questions going and some discussion. Kevin?
Thanks, MG. So I have, first of all, I am a pediatric cardiologist, so I am not a chemist or an engineer, so it's probably good that I just have one slide on nano. But for me, in my role in our Center for Pediatric Nanomedicine, co-directed with MG and others, and to really bring a clinical side to it. And that's how I first got interested in nanomedicine in the first place, and that's really just being in the cardiac ICU and getting tired of kids dying from heart disease, and why doesn't the damn heart squeeze? And, you know, the actin and myosin are there. And so, you know, can we work at that level, you know, really at the molecular level? And so that was really what pushed me to get involved in this field. And then my slide is when I think about as a clinician's view of nanotechnology and nanoscience and nanomedicine. And I think this is an enabling technology, and it's really a way to approach science. And for me, it's almost like a do-over. I mean, you can just start, you know, back, you know, investigation of all different diseases. And when I think of, you know, energy science, I mean, just approaching generation of power at the nano level. I mean, now we piezoelectric nano wires that are literally nanometer size that bend and generate electricity. And so there's power within the body, whether it's the respiration, whether it's just the blood moving around the aorta. If we think about devices that eventually will be coming that will need to be powered, just using nanotechnology to be able to harvest some energy would be fantastic. Physics, you know, there's a lot of quantum theory that really can be explored using nanotechnology and nanoscience. There's lots of wave particle theories that have been explored. Chemistry, since I'm sitting next to a world-class chemist, I probably shouldn't say much, but it really is, you know, if you think about, you know, working at the atomic level, I mean, that's what these guys do if you remember back to high school chemistry. So biology and molecular biology, a lot of that really can be looked at as nanotechnology and nano applications. But the cell can be described as the ultimate nanofactory. And if you have somebody with a disease of the mitochondria, and so we're looking at a patient in the hospital and you have mitochondrial disease and you're trying to, how can I improve this patient? Well, you're not even close to where the disease is actually taking place if the disease is within the mitochondria. And I think starting to be able to use these technologies to be able to visualize individual atoms and to be able to work at that level to be able to both diagnose diseases as well as find new therapies. And then material sciences are just, it's sort of another thing, it's almost like a do-over. So now you can design materials really at the atomic level. And so what type of atoms and relationships to whether it's blood or other materials that you're going to interact with. So the idea of heart valves that just do not, you know, become thrombosed. And if you have the right material on the surface, then blood platelets really will not stick to it. Computer science it's rapidly advances, obviously, but you know, we're now getting to the atomic level for storage of data. You can store data on individual iron atoms. And so it's just sort of massive new capacity that's going to be coming. And a lot of what Anthony and colleagues will talk about tomorrow about this whole data revolution, part of it is just massive data and data storage and acquisition. And so new techniques and technologies to advance computer science are going to be very, very important. And then all of engineering and biomedical engineering, there's plenty of you know, there's a tremendous amount of nano technologies and science that will be applied to that. And then I was trying to think of what analogy would I have to nanotechnology and nanoscience, to medicine, to research, to to innovation. And it's sort of one analogy I thought of, it's sort of like the development of the computer. And if you put the computer and computer science and data acquisition in the middle there, it impacts all areas of science and medicine. And I think that's what we're going to see with nanotechnology and nanoscience. Thanks.
So next one is Aman.
So really, my name is Aman Russom, and I'm from Stockholm, Sweden. And I could have talked using the former slide, actually, because I'm going to talk about nanotechnology, but specific for diagnostics, so point-of-care diagnostics. And as you can see from the title, it's going to be specifically designed to meet the requirements in resource-limiting settings. So I'm happy to be here today because when you think of neonatal diagnostics, the biggest group who are suffering from this one comes from resource-limiting settings. So if you look at the numbers, 98% of mortality come from poor countries. So, basically the question we have is, could a simple point-of-care test be of help to support children in the first 24 hours to 1 week, for instance? But let me take you one step backwards. When you talk about nanotechnology or microfluidics, I'm wondering if any one of you have seen this movie. Anyone? This really would show which generation the audience come from, because my students don't recognize it, I do so, which means, so this came from 1989, and really this professor, crazy professor, managed to shrink his kids, and which means when you talk about microfluidics and nanotechnology, this is when you shrink your scale dramatically, all the physics and behavior change. And we've been in this area where we call it nanotechnology, microfluidics, but lab on a chip. So when you talk about lab on a chip, it's shrinking a whole laboratory into a handheld device. We're getting there. So with this introduction, I would like to say in our lab, we've been working on different platforms to do lab on a chip. So as you can read here, I'm just putting 3 things: lab on a CD, lab on a foil, lab on a paper. And I think lab on a paper is easy to explain. If you think of pregnancy test, that is based on a paper. But the other ones, I have slides to show you. So if you think of a lab on a foil, it's like flexible newspaper, but in plastics. So what you can do with that one then is that you can have added biological functions into it as part of the production. So that would enable you to dramatically reduce the cost so you can use it at a resource limiting settings. So advanced biological diagnostics now could be done in this type of platforms. Another thing that I like, I think it was Wendy Su who was saying that they were hacking into the apps. What we've done here is, we call it lab on DVD, is that we hacked into a drive. So when you listen to music next time, think that you can actually do diagnostics with your DVD player. So literally what we have done is fill the DVD drive to act like a microscope. So imagine then while you can listen to your music, you can do diagnostics in the future. So what we're doing with those 2 platforms right now is we are collaborating with Gertrude's Children's Hospital in Kenya to actually look into point-of-care diagnostics for infectious diseases such as HIV diagnostics. But today what I want to also highlight is that we're looking into point-of-care for neonatal diagnostics the first week. We're working with a company called Kalmark in Stockholm, where basically we are developing a point-of-care device to look for LDH assay as a biomarker for severe disease. This is one of several biomarkers one can look into. So what it does is basically you take a plastic card, a droplet of blood, and then 3 minutes later you have an app telling you which concentration this is at. And with that, I would like to conclude to the younger audience, think outside the box. Thank you very much.
So I thought we had too upbeat of a conference, and I should really just bring us down a lot and talk about the controversies and challenges of nanoparticles and nanomedicine. No, I'm just joking. Actually, I think that there are actually really good opportunities, and if we can overcome these challenges or these controversies with nanoparticles and nanomedicine, it's going to open this huge floodgate of things so that our friends here and myself can hopefully really take on some of these really hard to treat pediatric problems, that really the only solution is nanotechnology or nanoparticles. So the first controversy that I think is useful to talk about is actually a little bit of the chemistry. And although I'm not a chemist now, I did major in biochemistry and I did work at Pfizer for a brief period of time. And so it's actually more challenging, well, maybe people do think it's challenging, but I think it's actually very challenging to figure out how to create the nanoparticle in such a way that it's application specific, right? So like MG was saying, it's not one size fits all. It's not, oh, it's a nanoparticle and one nanoparticle can treat every disorder. You have to design the nanoparticle in a way that it can deliver, for example, a drug or a light source or invade in a certain way. And then once you get, let's say you are delivering a drug, so it's a drug delivery nanoparticle, you have to maintain that the drug activity stays when it gets to its destination. And if it's a shuttle and it needs to come off, how do you get the particle off? And so those are all very interesting techniques, and I think maybe if we have some time, we can maybe talk about some of those challenges as well. And so if you think about delivery, it's not just delivery to that specific membrane to permeate into a mitochondria, for example. It's also about, for me as a neurologist, how do I bypass the blood-brain barrier? So that's eluded us in neuroscience. So for the past 20, 30 years, we've had very little advances in neuroscience in terms of penetrating the blood-brain barrier and delivering drugs effectively. And that's in many ways, you know, we see things that we've tested in mice, in cell lines, and then you try and get it past the blood-brain barrier, and the bouncers just won't let you in the club. So the nanoparticles might be our way to get into that really exclusive VIP club at Disney. I think it's called something 51, something like that. So I think that this is an opportunity more than a challenge, and it allows us to actually bypass some of the body's defense mechanisms that prevent us from delivering medications. And then the last one, and the one that I think is probably the most controversial and the one that's really going to challenge us to get past, is when we are introducing these nanoparticles, just like physicians are very resistant to change, I think individuals are very resistant to, are these going to be safe? Are there going to be long-term consequences to delivering nanoparticles, often which have cadmium, selenium, or other heavy metals, gold, for example? What are the long-term effects of these nanoparticles on the human being as well as the environment? So I think if we can look at these three challenges or these controversies and address them effectively, I think it will open the gates for us to really make a huge impact on pediatric innovation in nanotechnology. We won't go just yet to break, I think.
Thank you to our three speakers. I'll open it up in a minute, but let me start with a question or maybe a topic for discussion. And it's one of the things that I face in our own lab work, and I think the field faces. Because it is, in general, a platform technology, you have to choose to develop your own particular nanostructure for the particular target. The question is, what targets do we pick? And in a meeting full of folks who actually are on the front lines of caring for kids, the act of picking a target to take a run at with something like a nano approach is actually a significant problem that we face as a field. What problems do we choose? And so there are ones that perhaps are naturally amenable to nanoparticles, but others we know nothing about. So I'm going to direct this question initially at Kevin, who is, of course, a practicing physician and a very distinguished one, as to, is there a way that we should be communicating better with or how can we do that? How can we communicate better with docs to figure out what problems we should take a run at?
I think getting people in the same room is really the most important thing. And we've had this discussion in the past. Everybody is incredibly busy, whether you're, everybody tries to have a home life, or you have a very, busy clinical, work schedule. And then, can you actually get people in the same room that have the energy, the passion, and then the resources to make things happen? And I think, Anthony will tell you, it's innovation, but the next word that's part of that is collaboration. And hopefully that's really some of the excitement of, the I Spy type of thing. But for me is, just having an opportunity to be able to sit down and meet with people, brainstorm, come up with ideas, and there's just there is an infinite number of potential targets as well as applications that you could use, but it's always also a matter of coming up with the funding.
I should mention, with your work in the developing world, people might not know this, but nanostructures tend to be incredibly stable, very often very easy to produce, very easy to deliver. In many ways, a technology that is built for problems in low resource areas. And so again, the question is, do you see any particular opportunities there in diagnostics that you're not working on that the field might take a look at?
Yeah, I think we do use nanoparticles actually for enhancing signals in our fields, in our devices. So when you look into the, for instance, pregnancy test, it is a very useful platform, but not so sensitive, sensitivity-wise. So what we use nanoparticle for there is actually to enhance the signal. So using that, we can surpass some of the limitations. It's like having an amplification signal. So we do apply nanoparticles for point-of-care at resource-limiting settings in greater and greater phase right now. So that is one of the more exciting parts, is to take a biosensor that is made for a big, advanced lab into a field, and that's where the nanoparticles really are using, changing the way we do diagnostics today.
So knowing how I can go on and on, Anthony has told me we need to wrap it up. So Chi, do you have any final remarks? And then we'll open it up to one question from the audience, and then we'll have to move on.
Okay. We just talk about We just talk about the challenge. Okay, we have to get people together, and in an underdeveloped place, and we have to worry about how do we get the message through, and especially about the nanoscience or nanotechnology. The nano is about 10 to minus 9 order less than the human body. Okay, how can we get that a small material or small particles can be effective to influence our system, has efficacy with very low toxicity? That's one problem. And another problem is human problem. We get all the people sit together. First, engineers, scientists, and clinicians. The languages are completely different. Same word has different meanings. And that's the first hurdle. The second hurdle is after we learn how to translate all the languages among all the people sitting in the room, and then we have to give up some of our ideas which I learned since I'm from college, from each people, okay? So we have to go through all this process, and then we can get into the core of the problem. How can we use a nanoparticle or nanoscale medicine and then treat the patient? After that, then society problems. Okay, so really we're talking about how do we make a smooth transition from the length scale, from a very, very small molecule all the way to a society such that it will become effective to be applied to treat a disease, like a TB, like a HIV. It's not just a disease of a person, it's really society issues.
We have time for one question, I think. The magic box is back there. Go ahead.
Can you hear me?
I can hear you now.
You brought up a really important issue in regards to long-term consequences with this type of nanotechnology. Now that you brought it up, I'm struggling with the fact on how you make people comfortable with what you tell them in regards to long-term consequences.
You know, I think that's a hard thing. You know, if you think about GMO, for example, genetic modified food, or how we use antibiotics in food and our food sources and things like that, I think it's very difficult to find a balance between creating problems and helping the community understand. And I think it's open and honest communication and saying when you don't know what the long-term side effects are. I think the progression of nanoparticles in medicine is probably going to start, and it's already started with diagnostics. Imaging is probably going to be the next run at it, and then the last is going to be delivery of actual treatments and drug delivery. Although I will tell you that for terminal cancer for now, there's actually a clinical study that is starting, and they are administering the nanoparticles in those patients. And so I think it's if we're honest about, we have an option, this is a terminal illness, we have this solution, but we don't know if there's going to be long-term side effects of it. You know, I think that's important. But we've seen that in the cancer community before. You know, we started treating patients with, kids who had cancer with chemotherapeutic agents, and we lost their sperm and their ovaries, and we didn't think about that ahead of time. So, you know, but now we do that. We preserve their sexual reproduction. So I think honesty is really the best solution for that.
I'm really sorry we don't have any more time to go on, because this is, of course, a fascinating subject for all of us up on here at the stage. But let me thank our speakers and thank you for your attention.